Alright, so let's see if we can use what we've learned so far to talk about how COVID-19 replicates. At the beginning of this lecture, I talked about how there are a lot of different ways that viruses can have their genomes. They might have a genome that's based on DNA or one that's based on RNA. It might be linear, circular, single-stranded, or double-stranded. Based on the way the virus is structured, that's going to determine how its DNA is replicated and whether or not it goes through transcription before translation occurs. The most simple one to think about is double-stranded DNA viruses. Examples of this are adenoviruses and herpes viruses. These are going to pretty much work the same way you do. They're going to have regular double-stranded DNA, and that DNA is going to be replicated using a DNA polymerase that's DNA dependent, just like yours, and a DNA dependent RNA polymerase to make mRNA, just like yours, and it'll go through translation the same way you do. So these viruses are really not that different from you or I. The next option is single-stranded DNA viruses, like the parvovirus. These are also pretty similar. In this case, we're going to have one piece of DNA that's going to make a complementary copy. Now we have double-stranded DNA again, so everything is going to work pretty much the way it works in you. When it gets to RNA viruses, that's where things get a little bit tricky. You don't have any enzymes that are able to read RNA viruses and make replications of them, or make complements. So in order for an RNA virus to do anything with its genome, it has to bring with it a special RNA-dependent polymerase. In the case of double-stranded RNA viruses, it will behave exactly the same as double-stranded DNA would, except the virus will use its own RNA-dependent RNA polymerase to do everything that the DNA polymerase would be doing normally, and that the RNA polymerase would do for transcription. Single-stranded RNA viruses are a little different from that. They're still going to need to bring with them an RNA-dependent polymerase, but it matters whether or not the RNA is the positive strand or the negative strand, because your cells are only able to read positive strand RNA and translate it properly. This means that positive strand RNA viruses are able to go ahead and make proteins directly from their genomes. They're going to use the RNA-dependent polymerase to make a complement, negative RNA, that they'll then use to make more of their positive strand RNA to send on in their offspring. Negative sense RNA viruses make messenger RNA directly from their negative strand RNA using their RNA-dependent polymerase. They also make positive strand RNA so that they can make more negative strand RNA to send off in their offspring. This means that they have to go through both transcription and translation, whereas the positive strand RNA virus can go directly to translation. There are a few other types of viruses like retroviruses and adenoviruses, but today we're going to be skipping those. So which one of these is COVID-19? COVID-19 is a single-strand RNA virus, and it's positive strand. So this is essentially how it works. When the SARS-CoV-2 or COVID-19 virus comes into contact with your cells, it's going to bind to the receptor ACE2 on the cell surface. This is going to have the cell bring in the virus via receptor-mediated endocytosis. Once inside the cell, the virus will open and allow the RNA to enter into the cytoplasm. Some of that RNA will be used to directly translate viral proteins, and those proteins will be used to make a replication complex so that the RNA can replicate itself. Proteins and RNA that are made this way will then be assembled into a new virus in the Golgi and will eventually be released through exocytosis. So let's think about what is different and the same between you and the COVID-19 virus. Only one thing is actually the same, and that's the way translation works. So messenger RNAs, or single-stranded positive strand RNAs, are read by ribosomes and translated using the same codon chart. Everything else, on the other hand, is different. The viral genome is RNA, whereas yours is DNA. It's single-stranded, not double-stranded like yours. Single-stranded RNA genomes are directly translated, if they're positive strand, and no transcript is made first. And the RNA is replicated by proteins that are encoded in the viral genome, because it needs an RNA-dependent RNA polymerase. So as you can see, viruses like double-stranded DNA viruses are pretty similar to you, but viruses like COVID-19 are very different. It's important to understand how a virus replicates to try and interfere with how it might grow, and thus find a way to combat it.